And wonderful panel of speakers. We have Anais Franco Romero joining us from University of Padova, Italy. And she's going to be talking about the novel uncategorized gene MYTHO, which controls autophagy and longevity. Anais, please go ahead. And I apologize if I'm mispronouncing your first name.
It's fine. Can you hear me?
Yes, you can. We can hear you, and you have 10 minutes.
Okay.
If you can leave just one quick minute for a Q&A, that would be great. Thank you.
So, hello everyone. It's really a pleasure to be here and present you my data about a novel uncharacterized gene that we found is Foxo-dependent gene that regulates longevity. Can you see my slides here? Okay.
Yep, perfectly.
So, healthy aging, as you know, is a very complex phenotype, which is influenced by environmental and genetic factors. So doing exercise and eating well, it's good to improve health span. But the point is that during aging, the genetic contribution increases even more. That's why a lot of efforts are trying to elucidate key pathways and key genes that could be prolonging longevity or decreasing the aging decline. So I'm going to talk about among all the hallmarks of aging, the regulating nutrient sensing is the most, was the first to be described to regulate aging by the insulin signaling pathway. This is a very conserved pathway, which is activated by insulin and growth factors, which at the end activates AKT, and it can phosphorylate and prevent Foxo transcription going into the nucleus and transcribe the genes. So when this pathway is inactive, now Foxo is free and goes to the nucleus and activates transcription of genes. Among these genes, there are a lot related with longevity. So for example, as you know, when we mutate DAF2, which is the receptor for insulin in C. elegans, we can prolong longevity. It was reported. And when we double mutate Foxo together with DAF2, you can actually reduce the longevity of DAF2 animals. So it's important, this pathway, to regulate longevity. But which are the genes that are regulated by Foxo? So there are a lot of different genes. Some of them are directly regulating the detoxification of the cells, so they reduce ROS levels. Also, there is some genes related with mitochondrial function. Genes also involved in the stemness, and importantly, genes directly involved in proteostasis. So this means that it regulates genes that have a role in the two main proteolytic systems that we have in our cell, which is the autophagic lysosomal system and the ubiquitin proteasome system. So of course, low proteostasis would lead to an increase of cellular damage because there is a lot of cell damage organelles and misfold proteins. So just this slide to remind you that even though Foxo has a role in this hallmark of aging, it's also influencing other hallmarks. For example, the loss of proteostasis, mitochondrial dysfunction, also the stemness of the cells, and cellular senescence. So our lab was looking of genes that anyone has studied them, because there are actually 40% of the protein encoded in eukaryotic are proteins that doesn't have any function because anyone is studying them. So we wonder which genes under Foxo control, other new genes, could be influencing longevity. So we came out with, we had microarray data, we did perform a microarray data from muscle biopsies, comparing wild type mice with knockout, Foxo knockout, in fed and starved condition. And we look for genes that were upregulated in starvation, where we know it's a catabolic condition where Foxo is very active, and it's totally depleted in the Foxo knockout mice. And among these, we found one very interesting gene that was very conserved between in C. elegans to mammals. And it's a RICAN gene, which was annotated by the RICAN Consortium database. And there is no publication about that. So even though the microarray was performed in muscle, it's not a muscle-specific gene because we observed that it was transcribed in a lot of different organs, mostly in the liver and the lung. Also, protein expression seems to be increased in the lung and in the liver more than in other organs. But is mito induced in aging? So we found that in a group of mice of 2 years old, they were highly increased compared with the younger ones. And not only mice, we have analyzed a group of patients and we found higher expression of mito in the group of patients from 84 to 95 years old. So it seems to be an age-dependent gene. But to see how this mito was influenced into aging, we actually created a CRISPR-Cas9, by CRISPR-Cas9, a C. elegans model, and we depleted this gene and observed that there was a reduction of lifespan. But is mito regulating the effect of Foxo on lifespan? So we created a double mutant together with DAF16, and we found that it was decreasing the maximal lifespan, but not the mean of lifespan. And together also, DAF2 and mito knockout did not reduce the lifespan of DAF2 animals. So we suggest that mito is not mediating the Foxo effect on lifespan, but it's doing it by another pathway. So probably it's another, in basal conditions, other pathways are regulating mito and controlling longevity by itself. So then we analyzed if we give an oxidative stress, what was the survival of these animals? And it seems to be decreased when we give paraquat, that we know is increasing ROS production. So they were less resistant to oxidative stress. Then also we analyzed how was the health span, and we found it decreased it was decreased the number of body bends and the number of reversals, meaning that it wasn't impaired a motor function. So we have another project going on that we know mito is controlling autophagy, and we know that mito is controlling a lot of autophagy genes. So we wonder how was autophagy in this C. elegans model. And we found that the autophagosome formation, it's impaired compared with the wild type. And in parallel, we created another model in zebrafish where we depleted exon 5 of mito, and we also reduced the lifespan of these animals. And also motor function seemed to be impaired. So why mito knockout age? Which is the mechanism that mito is controlling aging? I'm sorry to tell you, I don't have the answer yet, but we have several hypotheses and several projects going on, because we think it has also going with a mitochondrial alteration. We observed this very strange mitochondria swollen, and also mitochondrial ROS production, it's increasing the knockout compared with the wild type. So it could be contributing to aging. On the other hand, we have a project also where we found that mito is directly interacting with WEP2 and ATG7, so some proteins of the autophagy machinery. And these animals had also a decrease of autophagic flux. These cells, C2C12, were also, they had impaired autophagic flux. And also in the model we have in zebrafish, we observed the same. So we think it's because of this reason. So the summary of this talk is that we discovered for the first time a new Foxo-dependent gene that was upregulated in aging, that mito knockout in C. elegans and zebrafish had a reduced lifespan, and it seems to have a problem, a motor function impairment and low resistance to oxidative stress. And the mechanism, it could be related to autophagy impairment and mitochondrial dysfunction. So I would like to thank all my lab, especially, of course, Professor Marco Sandri and all the collaborators, Valeria Morbidoni and Filippo Citon, Eva, and also Luisa Della Valle. And thank you all for your attention.
Thank you very much. Thank you very much. Really a great talk. We have time for one question from Joris Steelen. Are there known genetic variants in MYTHO that are influencing age-related phenotypes in humans?
So if it was, can you repeat the question, sorry?
Are there any known genetic variants, or are there SNPs or mutations that lead to a phenotype in humans?
This is a very interesting question. We look at it, but it doesn't seem to be like that, actually. Yeah, we look at it, but not. I'm sorry for that.
No worries. And there will probably be other questions online, so I really highly encourage you to go to
Yes,
Slack.
sure.
And as you know, we have a very tight schedule with these short talks, but sincerely appreciate your talk. And
Thank you.
please go on Slack.